Hello, my name is David Roos, and I'm a professor of biology at the University of Pennsylvania and the Penn Genome Frontiers Institute in Philadelphia, USA. And it's my pleasure to be here with you today, aside from the fact that I'm getting over a head cold, which I hope doesn't make my voice too congested to be intelligible, because it's exciting to be able to talk to you about the new discipline of genomics, which I understand from my colleagues John Hoganesh and John Murray that you've heard quite a lot about over the course of this lecture series. But I'd like to talk to you today about a slightly different aspect of genomics, the genomics of pathogens, which differs only slightly from the genomics of humans and model organisms that you've heard about before. In the same way that you've heard over the course of previous lectures, we're interested in the genomes of these organisms, the expression profiling, the imaging data, and all of the other very rich data sets that you've been hearing about all course long. But in contrast to the application of those genomic problems in genetic disorders and developmental disorders, where it's often very challenging to try to even identify the basis of the problem, and much more challenging to try to correct it, our goal in pathogen genomics is in some ways a much simpler one. Our goal is usually to try to kill these organisms, which are the source of much human disease and suffering. And because our goal is simply to try to kill them, we usually care quite a lot about what the organisms do a lot of, and that's often easier to address, and I'll give you some examples of that. Today's lecture will be divided into 4 segments: an introduction to the biology of malaria caused by the Plasmodium species responsible for this disease, an introduction to the genomics of these organisms, a discussion of the discovery and characterization of the apicomplexan plastid, or apicoplast, and a more general discussion of the use of genomes to identify therapeutic targets and moving towards the development of new drugs and vaccines. And each of these sections will be associated with a series of questions at the end. Because the lecture is a little bit long, in order to try to minimize the bandwidth for downloading, we'll break it into 2 sections, but it's all part of one presentation. So let's get started. Malaria is a serious disease that is caused by species in the phylum, in the genus Plasmodium. And while we don't hear much about malaria in the U.S. or in Europe these days, because it has been eliminated over the preceding century through a series of public health measures and drug treatments and other interventions, malaria is still very much with us and still very much a problem in much of the tropical world, particularly in Africa, South Asia, and in parts of Amazonia, as you can see from these maps produced by the Malaria Atlas project that is accessible online. Still very much a problem, depending on who you are and where you live. The reason it's still a problem is that, while we can see a dramatic decrease in global malaria over the period from the 30s through the 60s, this control bottomed out in the late 60s and early 70s, largely or because of a variety of reasons, economic reasons, political reasons, the development of resistance on the part of both the parasites and the insect vectors that transmit them. We can see that in the graph over at the left, where chloroquine, which was once the mainstay of anti-malarial drug development by the time this graph was initiated in 1976, was of greatly declining efficacy, to the point that in much of the world it is now essentially useless. That led to the application of a combination of antifolates, sulfonamides, and pyrimethamine, which were once very effective but have been of declining efficacy, such that by the mid-'80s, these drugs were again not very useful in much of the world. Other antimalarials are also available. Drugs such as mefloquine, for example, were introduced in the mid-'80s, but you can see the declining efficacy of mefloquine over across much of the world. And even quinine, a drug whose mechanism of action is still poorly understood, but which was the first antimalarial identified as a natural product in cinchona bark, is of declining efficacy. As a result, current therapeutics therapy for malaria relies on another remedy derived originally from an herbal medicine, artemisinin compounds based on Chinese herbal remedies, are now the mainstay of antimalarial treatment, and there have been reports of treatment failures that are very worrisome. You can certainly see that the dramatic improvements in antimalarial control over the 50s and 60s were halted and even reversed in the 70s, most notably in Africa, where malaria is on the rise. This disease is caused by parasites in the genus Plasmodium within the phylum Apicomplexa, a large group of eukaryotic pathogens comprised of more than 5,000 named species, all of which are parasites. And this slide makes the point that among the great domains of life, the bacteria, the archaea, and the eukaryotes, while most of the microbial diseases we are most familiar with fall in the bacteria, even within the eukaryotes, the nucleated cells, the vast majority of eukaryotic life is microbial eukaryotes, indicated in pink. Humans are just a twig out on the end of this branch, and even animals, plants, and fungi are relatively recently emerging branches on this tree of life. Among the microbial eukaryotes shown in pink are many, many thousands of species, several of which we care quite a bit about as the cause of human disease. The Apicomplexa are a phylum indicated in red, and just as you've heard that that model organisms have been very informative for understanding the biology and development of humans, various species of apicomplexans can serve as models for each other. On the right are indicated several genera of apicomplexan parasites, including the Plasmodium species that cause human malaria, the Toxoplasma parasites that cause toxoplasmosis and congenital disease, and diseases of immunosuppressed patients in humans and various animal species, the Cryptosporidium parasites that are also responsible for disease in immunosuppressed individuals, Theileria parasites, which cause a fascinating and devastating lymphoma in cattle. And while these organisms have very different life cycles, Plasmodium parasites are carried by mosquitoes, for example. Toxoplasma parasites are carried by cats. They are closely related evolutionarily, and we can therefore use one as a model for understanding the biology of others in ways that you will see in a moment. Let's take a look at the biology of Plasmodium, a fascinating topic that I won't be going into in detail. I direct you to the reference shown on your screen, or on many other references that you can find online if you're interested in more about the biology of malaria parasites. Malaria parasites are carried by mosquitoes, and when a mosquito bites a human, when an infected mosquito bites a human, it releases sporozoite stage parasites, long, slender parasites that move through the blood and in the space of minutes enter into the liver, where they invade hepatocytes and transform themselves into a completely different looking organism with very different biology. And this is one of the challenges of dealing with microbes, in particular eukaryotic microbes of this degree of complexity, that change themselves into a very different form. In fact, it took many, many years before biologists even discovered that the parasites observed in the gut of mosquitoes were responsible for the disease observed in the blood of humans. In the liver, over the course of several weeks, malaria parasites transform themselves into yet another form. They replicate and burst out of liver cells in the form of merozoites that infect red blood cells in the circulation. And we will be talking today solely about the intraerythrocytic cycle, the human blood cell cycle of malaria, because this is what's responsible for all of the pathogenesis in humans. Merozoites invade red blood cells and transform themselves into yet another form known as ring stage parasites, because they look a little bit like a ring here. They invade into cells, red blood cells, a very inhospitable home which is not used by very many pathogens, where they establish an intracellular home and gobble up the hemoglobin inside the red blood cell, degrading the protein as their source of nutrients, polymerizing the heme into essentially an indigestible rock known as hemozoin, and proceed to grow in the form of trophozoites, indicated here, with the hemozoin polymerized in a waste product that will be left behind. Having exhausted the resources of the red blood cell, the parasites transform themselves yet again into another form known as schizonts, replicative parasites in which they assemble daughters essentially de novo within the mother, which burst out as merozoites again. And you can see multiple merozoites here released from malaria parasites, leaving behind this indigestible polymerized heme as hemozoin. This human red blood cell cycle can proceed over many phases, leading to anemia, leading to many sorts of other disorders in the blood. The parasite-infected red blood cells themselves can arrest and get clogged within the arteries of blood vessels of the brain, causing miniature clots and diseases similar to stroke. Many sorts of symptoms that can lead to the devastating biology of Plasmodium falciparum malaria, the most lethal form of human malaria, and other aspects and the other forms of other biological ramifications in the other forms of other species of malaria that give Plasmodium give rise to human malaria. Some of those parasites will differentiate yet again into another form, in fact, two different forms, male and female gametocytes, boys and girls, which are taken up by the mosquito, and in the gut of the mosquito then undergo sexual recombination differentiation through a series of other forms that we won't be talking about today. Again, a fascinating form of biology, but those parasites undergo sexual recombination, whereupon they produce new sporozoites that migrate up to the salivary glands, where they're available to be injected in the next blood meal taken by malaria parasites. Now, these organisms, these eukaryotic cells, have nuclei, and those nuclei harbor DNA, and that DNA is a DNA genome, very much like the genomes that you've heard about for other organisms. It consists of a variety of chromosomes, 14 chromosomes in malaria parasites. All right, here are 14 chromosomes in malaria parasites. Those individual chromosomes, like chromosome 4, shown here, consist of many hundreds or even thousands of genes, and those genes are similar to but different from one species of malaria to another. Here, Plasmodium falciparum, the most lethal form of human malaria, Plasmodium vivax, which is perhaps even more widespread but less commonly lethal, and other malaria parasites such as the rodent malaria parasite Plasmodium uelii, indicated here. We can see that many of these genes are conserved between species, but not all of them. In the telomeric regions, we see a variety of genes that are distinct to different parasite species, and there are other genes, including amplified clusters of genes, the variant surface antigens that are responsible for, or thought to be responsible for sequestration in the tissues for Plasmodium falciparum. Other genes that are amplified in Plasmodium vivax, such as these Vir genes as indicated here, that are also the subject of great interest. We can see that the science of genomics allows us to be able to gain some insight into the function of these by looking at orthologous genes across species, and that even for genomes that are not very well assembled, as indicated by the many fragments of genes in the Plasmodium yoelii genome, we can still learn quite a lot about many of the proteins genes in these parasites and the proteins they encode. Those proteins can be assembled into putative metabolic pathways by analogy to other organisms. We can see here the metabolic pathway of glycolysis, where glucose is converted into pyruvic acid, how acetyl coenzyme A in the mitochondrion is used through the Krebs cycle for the production of ATP, the energy component of the energy requirement for cells, when we can also see various parasite-specific functions, such as the digestive vacuole here that's used for the degradation of hemoglobin, and an intriguing organelle known as the apicoplast that we'll be talking about in the next section of this lecture. So that brings us to our first set of questions. Question set 1 about malaria and pathogen biology. When was malaria eliminated? In the US, in Europe, and in Africa? So 3 parts, I guess, to this question, 1.1 A, B, and C. A, in the 1800s, B, in the 1940s, C, in the 1960s, D, in the 1980s, or E, has malaria not yet been eliminated? And again, this is 3 questions: the US, Europe, and Africa. Question 1.2. What factor or factors might pose a serious challenge or challenges for malaria eradication? The emergence of drug resistance in malaria parasites, the emergence of insecticide resistance in the mosquitoes that carry malaria, the complicated life cycle of these parasites, any 2 of the above, or all 3 of the above. Question 1.3. Among the following species, Plasmodium parasites are most closely related to humans, fungi, plants, bacteria, or are they equidistant to humans, plants, and fungi? A is humans, B is plants, C is fungi, D is bacteria, and E is equidistant to humans, plants, and fungi. And finally, some thought questions that we won't be answering, but which you should keep in mind through the remainder of this lecture. What features of parasite biology are unusual, by which we mean distinct from more familiar organisms, such as humans? And what criteria might be most useful for identifying candidate therapeutic targets in the parasite genome? Give you a moment to answer those questions, and we'll continue with the rest of the lecture. In the second part of this lecture, I'd like to tell you a specific story about the discovery of an unusual aspect of malaria biology that has proved to be significant from a chemotherapeutic standpoint. And this story got its start from work by Maria Fiqueira when she was a graduate student in my laboratory, in which she sought to identify why these antibiotics shown at the top of the page, structurally dissimilar antibiotics, but which many of you are familiar with. You may have taken them to treat an ear infection as a child, or to treat a Helicobacter infection that is giving rise to incipient ulcers, or many other bacterial diseases. But as I've already told you, and as you've already seen from the evolutionary tree that we looked at earlier, malaria parasites are not bacteria, and one might therefore be surprised that these antibacterial antibiotics, which after all kill bacteria but not eukaryotes, such as humans or malaria parasites, you might be surprised that these antibiotics would kill malaria parasites, but indeed they do, and by a very peculiar means that I don't have a time to go into, but which you're welcome to look at in the references cited. And through a long series of studies, we and others identified the only plausible target for these antibiotics as a previously mysterious episomal, that is to say, non-chromosomal DNA in Toxoplasma and Plasmodium parasites. This DNA was not shown on the 14 chromosomes that I showed you earlier from Plasmodium parasites. It's a small 35,000 nucleotide circular DNA that was originally assumed to be the mitochondrial genome of these parasites. But when the true mitochondrial genome was discovered, that left this episomal DNA as something of a molecular biological mystery, a DNA without a home or without a function. But because the ribosomal genes encoded on this circular DNA are plausible targets for macrolide antibiotics and lincosamide antibiotics, such as the clindamycin and the zithromycin and chloramphenicaldrix that I showed you before, which might be expected to target the protein synthesis on the large subunit ribosomal RNA, we were interested in exploring whether this might prove to indeed be the target for such antibiotics in these parasites. Now, having identified this as a target for these antibiotics made it much more than a molecular biological mystery, and a mystery that it was important to try to define as whose function will be important to define. And we can use the genes encoded by this genome, not only the ribosomal genes, but genes predicted to encode ribosomal proteins indicated over in this region, or genes such as elongation factor TU, which is very well conserved across evolution to try to identify the origin of this mysterious episomal DNA. And the molecular phylogeny, the evolutionary tree indicated it right, gives us an answer to this question. It tells us first and foremost that the phylogeny of this episomal DNA in Plasmodium is closely related to Toxoplasma and Eimeria, another veterinary pathogen of importance in the poultry industry. It tells us that it bears indeed no similarity to mitochondrial genomes, which are in fact of bacterial origin, and hence as we know from many studies over the years. It tells us in fact that they are much more closely related to the genomes of cyanobacteria, the ancestor of chloroplasts in plants and algae. But what was really quite a surprise was that in fact the apicomplexan plastid, or apicoplast as it has come to be known, emerges not as an independent colonization from a cyanobacterial or other bacterial ancestor, but grows out of the tree from the plants and algae themselves. If we look at the diagram of an evolutionary tree over at left, which symbolizes much of what we know about eukaryotic evolution, we know that mitochondria arose by an endosymbiotic colonization when an ancestral alpha proteobacterium invaded the ancestor of all eukaryotes, although some eukaryotes, such as the pathogen Giardia, have subsequently lost their mitochondrial genome. Similarly, we know that the origins of plants and animals, the origins of chloroplasts arose when a cyanobacterial ancestor colonized the ancestor of all plants and algae. But the placement of Plasmodium in this tree, emerging as we see in the tree at right from the plant and algal clade, argues for a secondary endosymbiotic process. That is to say, the ancestor of these parasites were invaded by, or ate, depending on your perspective, a eukaryotic alga, which had previously acquired its plastid by that primary endosymbiotic transfer from a cyanobacterium. It's a little bit confusing. Here's a cartoon diagram illustrating the same process. The origin of the chloroplast when an ancestral eukaryote, with its nucleus indicated here, ate or was invaded by a cyanobacterium, giving rise to the chloroplast surrounded by a double membrane. And now in pink, along comes the ancestor of these parasites, or an ancestor of these parasites, which ate a eukaryotic alga, retaining the algal plastid, which we might expect would be surrounded by a complicated membranous structure. If this were the case, we ought to be able to see that organelle, and indeed we can, shown here not in Plasmodium parasites, but in Toxoplasma parasites, which are a more experimentally accessible organism, certainly exhibiting better ultrastructure. We can see in this fortuitous electron microscopic section an image that identifies an organelle distinct from the parasite mitochondrion, nucleus, or other intracellular organelles such as the Golgi apparatus. And indeed, in situ hybridization experiments have labeled this organelle as the definitive home for the apicoplast genome. Here in Plasmodium parasites, the nucleus indicated in yellow, the Golgi apparatus indicated in purple, the specialized secretory organelles that this parasite uses for invasion, this particular one known as the rope tree, the mitochondrion indicated in red, and the apicoplast indicated in green of this Plasmodium knowlesi, parasite. Now, the big question, of course, is what is it that the apicoplast does? Here in another electron micrograph of Toxoplasma, we can see the apicoplast, and this is now not just a molecular biological curiosity or a cell biological curiosity or a pharmacological curiosity, but a validated drug target, a validated therapeutic target, the target for chloramphenicol or clindamycin, which is used clinically to treat toxoplasmosis. If we could identify what this organelle does, we can perhaps identify new drugs to deal with the growing problem of malaria and the emergence of malaria, of resistance to existing drugs. But here, unfortunately, are the apicoplast genome, which was so informative from the standpoint of understanding the location and importance of this organelle and replication of this apicoplast genome, transcription into proteins, into RNA, and translation into proteins. The metabolic functions are essentially a mystery, no clue from the apicoplast genome. And that is presumably because most of the functions of this organelle, as indeed most of the functions of the chloroplasts in plants or the mitochondria in most eukaryotic cells, are encoded in the nucleus and post-translationally imported into the organelle. How can we identify what those functions might be? And to answer this question, we turn to the parasite genome. Initially, random cDNA sequences, expressed sequence tags, or ESTs, from the Toxoplasma and Plasmodium genome, and as genome sequences emerged over the past decade or more, the genome sequences of these parasites themselves. And we could look in those genomes, for example, for ribosomal proteins necessary for function, but which were not encoded by the ribosomal protein genes of the apicoplast genome, as indicated in yellow. And here we can identify some proteins such as ribosomal small subunit protein S9, without which you can't make a functional ribosome, or the large subunit protein 28. And antibodies to these proteins identified proteins on Western blots from Toxoplasma and Plasmodium parasites. And what's more, those proteins in the genome were predicted to contain a long N-terminal extension, which you might imagine would be involved in targeting to the organelle. And on Western blots, they could be identified as having what looked like both precursor and mature forms of those proteins. This from work largely done by Ross Waller and Jeff McFadden's lab at the University of Melbourne. We can use those antibodies to identify the apicoplast itself. Here in Toxoplasma parasites, we can see that indeed immunofluorescent staining labels the apicoplast and organelle distinct from the nucleus, shown in red in these images. And by electron microscopy, we can once again see the apicoplast as distinct from the mitochondrion or the nucleus of these parasites. Similarly, in Plasmodium, in this case by labeling with a fluorescent protein reporter, we can see the parasite as it transits from those ring stage parasites to the trophozoites and on to schizont stage parasites that now must assemble multiple daughters within the infected red blood cell using this bizarre snake-like structure before assembling here 16 daughter merozoites that will burst out of the parasite, leaving behind that crystal of hemozoin as they go on to infect new red blood cells. Because we now know that this long N-terminal extension is likely to mediate targeting to the apicoplast, we can test that hypothesis by truncating that N-terminal extension, removing that N-terminal extension and fusing it to a fluorescent protein reporter such as GFP, and following that targeting to the apicoplast. And here, in work carried out in my laboratory by Boris Streepin and later Omar Harb, we can see the targeting of this targeting signal indicated in this case in blue and green properly targets a fluorescent protein into the apicoplast. When we remove one piece of, when we truncate part of that protein, we see that in fact the protein remains within the parasites. When we truncate other parts of the protein, we see that they're secreted outside of the parasites. Then we can use these proteins through more extensive cut and paste molecular genetics to distinguish between the proteins, the parts of the targeting signal that are responsible for trafficking to the membrane or into the lumen of the apicoplast. In summary, we now know that targeting to the apicoplast involves a remarkable bipartite targeting signal, a pink bit here which mediates co-translational translocation across the endoplasmic reticulum membrane, whereupon signal peptidase cleaves that signal sequence, exposing the yellow bit, which is in fact a chloroplast targeting domain, betraying the plastid ancestry of this organelle. That yellow bit now mediates translocation across the remaining membranes into the apicoplast, whereupon a plastid processing protease removes the yellow bit to leave the mature protein that remains within that organelle. And that brings us to the second set of questions for this lecture. Question 2.1. It is surprising that drugs like clindamycin kill malaria parasites because A, Plasmodium is a plant, B, these drugs are antifungal agents, C, these drugs are antibacterial agents, D, these drugs do not enter into red blood cells, or E, malaria parasites are resistant to known drugs. Question 2.2. Why does the phylogeny of apicoplast genes suggest secondary endosymbiosis? Is it because A, they're related to mitochondria, B, they're related to toxoplasma, C, they branch off of the evolutionary tree from the algal lineage, D, they branch from the tree out of the cyanobacterial lineage, or E, they are unrelated to anything else that we know about. Question 2.3. What does the apicoplast genome encode? Does it encode A, machinery for RNA transcription, B, enzymes for protein translation, C, metabolic enzymes, D, two of the above, or E, all of the above? And finally, a short essay question, question 2.4. How does the distinctive bipartite apicoplast targeting signal mediate trafficking of nuclear-encoded proteins into this organelle? I'll give you a few moments to think about that, and we will move on to the third part of this lecture, which will be in a different file to download in the interest of saving bandwidth.